Okay, here we start chapter 18. In the chapter 18, we are going to see cell division cycle and mitosis, which you have already seen actually several times in your life before. Well, we are interested in cell cycle mostly because of for the cancer features and to study cancer, but it's actually everywhere. All the eukaryotic organisms you see around or you don't see around, they share the same mechanisms of mitosis. So this is quite a shared thing. And we know that all the cells are coming from the previous cells, so they have all came to life with the same principle. So for any kind of cell, there are two phases. First, there is a cell. Okay. This is the cell living, and then the cell grows. It should increase its content, and then it's going to divide into two. Okay. This one is called the parent cell, and these are called the daughter cells. Okay. This part of the cell cycle is called the M phase, and this part is called the interphase, which is the growth phase. So many things are going in the growth phase, but if you check under the microscope, it's going to look mostly dull, mostly silent, though all the biochemical things are happening there. And the M phase is actually divided into two. There is the mitosis, which is the division of the nucleus, and there is the cytokinesis, which is the division of the cytoplasm. So maybe you have been using mitosis instead of cell division or proliferation, but now you know that mitosis implies only and only division of the nucleus. So the interphase or the cytokinesis, they're not part of mitosis. And well, we call it cell cycle because every cell goes through in this cycle. Okay, so it grows, it replicates its material, and then divides, and then grows again, and then divides again. So many cells can divide several times. Many cells can make copies of themselves so many times. If you're talking about a cell of a multicellular organism, well, they have limited lifespan, limited number of times. But if you're talking about the unicellular eukaryotic cells, they can divide indefinitely, and that's how they reproduce actually. Okay, and you're going to see this is the interphase. This is the interphase. No, this is the interphase, and then this is mitosis plus cytokinesis. So this is the M phase. Okay. Sorry, my handwriting is really bad. Okay, this cell cycle can take really different times between cell to cell. For example, if you check the egg embryo right after its fertilization, they are dividing in every 30 minutes. It's quite fast. And if you think about the yeast cells, it's 1.5 hours. And if you think about the cells in your intestine, which are rapidly reproducing, it's half a day. And the fibroblast cells in cell culture, it's approximately a day. And if you check other cells in your body, it can take 3 days, 1 month, 1 year, more than that. So the cell cycle can change a lot between cell to cell. This is not because this division, this M phase is different. This M phase is quite fast and quite stereotype, while the interphase can change a lot between cell to cell. A cell can take quite a lot of time to grow, so this growth, this replication of material can take more time. It can stay there, it can wait there, but after the interphase is completed, this division into two, which is mitosis plus cytokinesis, is really fast. Okay, and we know that in the interphase is actually divided into three phases: G1 phase, which is gap 1, S phase, and G2 phase. Okay. Again, if you check it under the microscope, you wouldn't see many things in G1, S, G2, but it's extremely important. Let's start with the G1 phase. The G1 phase is the production phase. See, the cell is growing. Growth means synthesis. So what does it synthesize here? If a cell is going to grow, it's going to synthesize certain molecules. First of all, it should synthesize DNA. The DNA should be replicated. And as you know, DNA replication is very well controlled. It is not like replication of other things. It is not like doubling of other molecules. DNA replication should be perfect. And then the other materials like protein, lipid, carbohydrates, glycosylated proteins, organelles, they should all be replicated so that the cell will grow. And it's going to happen in the interphase. In the G1 phase, more material is done, more material is synthesized. In the S phase, it is specifically DNA replication. And in the G2 phase, it can wait or it can do some controls. And then the M phase starts. Okay. So, of course, the cell cycle should be controlled very well. Because we know that if there's anything going wrong in the cell cycle, what you are going to see is a tumor cell. In order to prevent tumor, what you have to control is the cell cycle. So in the cell cycle, there are some certain checkpoints. So checkpoint is really like a border. Imagine there is the Turkey here and Greece here, and there's a border between them. So to pass from Turkey to Greece, you have to show the passport. Okay? Unless you have the proper passport and proper visa, you cannot enter into Greece. And this is a sharp passage. I mean, the border between Turkey and Greece, it is very well defined. It is not blurry. So it is very well known who is passing the border and who is not passing the border. And it is very well known in what circumstances the people are going to be allowed to pass and in what circumstances they are not going to be allowed. So these checkpoints, one is here, one is here, and one is here, they are like borders between countries. So in that case, you leave one stage and you start with the other stage. The passage between them is really sharp, well controlled, and the next one will not start before the previous one finishes. Okay, this is required for a healthy proliferation, for a well-controlled proliferation. Okay, it checks few things before starting the S phase, passage G1 to S, it checks the environment. From G2 to M, it checks the DNA, and in the M phase, it checks the mitotic spindle assembly. To be more specific, here between G1 to S, what it checks is the environment. It checks if there are social signals. Now remember, there must be social signals called paracrine signaling, maybe sometimes hormones as well, which is also called endocrine signaling. Because if cell division is not requested in a body, individual cells should obey this rule. So they should talk to these body centers, they should talk to their neighbors if they are okay with the cell division. If they are not okay, the cell division should not continue. Okay? These are some outer signals. These are outer information. Okay? In addition to social signals, it should check if there is enough food. Because if there is not enough food for one single cell, there is no point of cell division and to make two cells, because it obviously will not be enough for two cells. If there is enough oxygen and if there is enough space, because if the environment is overcrowded, it's not going to divide. I mean, there should be enough space for a new coming cells. Okay, and there are some inner information. This inner information is about the current condition of the cell. For example, it checks the cell size. It should be big enough to create two cells because you're going to divide into two and it's going to become 1, 2 daughter cells. If it is not big enough, for example, if this cell divides without any growth, after division, the daughter cells will be too small and it's not welcome. This is going to be a problem. So we see this kind of cell division without any growth very rarely. Normally, in a healthy cell, it should grow, double the content, and then divide into two, and this should not happen. Otherwise, the cells would become smaller and smaller. So it's going to check if the cell size is okay. And then it's going to check if there's any damage in the DNA. So if there is any kind of mutation, it should be repaired. So for example, here there is a mutation detected. The cell cycle will stop here, okay? It's not going to continue until this mutation is repaired. So maybe it's a small mutation, it can be repaired by the DNA repair system easily, so it can continue. Maybe it's something bigger, it's going to take more time. Maybe it's a chromosome breakage. Maybe it is so big that it's not going to go to the next phase at all. It's going to stay in G1, or maybe it's going to stay in G0 forever. It will just leave the cell cycle. Or maybe the damage is so big, it will just suicide, because not every cell is meant to survive. Okay, so this is what it checks in the G1, and we say this is the main border, because once it passes the G1 to S, the cell cycle will continue and come back here, sooner or later. Okay, so this is the main thing. The cell decides proliferation here. If it passes here, it proliferates. If not, it's not going to proliferate. Okay, another checkpoint is here at the end of G2. Here, it checks the inner information. Of course, the DNA is checked always. Again, if there is any mutation in the DNA, the cell cycle will stall and wait until the DNA damage is repaired. And the second thing about the DNA is it checks if the DNA is replicated. Because every piece of DNA should be replicated, every single nucleotide, and they should be replicated once only. So if there is some chromosomes stay unreplicated, the cell cycle is going to wait until everybody is replicated. And finally checks the MPF, which I'm going to describe soon, which is the maturation promoting factor. And here, in the middle of the M phase, which is actually right after the metaphase, it checks if the chromosomes are attached to the spindle apparatus. Because if they are not attached, they will not be able to segregate. Okay, and then if everything okay, it divides into two, mitosis, cytokinesis, and it creates two different cells. Both of them are at the G1 phase. Okay, how the cell cycle is controlled, let's